Bismillahirrahmanirrahim. Welcome to Botanica. So we have almost completed our syllabus of plant tissue culture. We have discussed the most of our topic, or we have completed 80, 85 to 90% of our syllabus, and there are some small topics which we are going to study in this week, inshallah, and we are going to complete it. So we have completed the in vitro pollination, its application, why there is a need of in vitro pollination, what is the process of in vitro pollination, methods used for in vitro pollination, and now we are going to discuss about and we are heading to a next topic, that is the germplasm conservation. So as far as the germplasm conservation is concerned, we study this particular topic in the genetics, in the biotechnology, and in the tissue culture as well. And why we are going to discuss it in the tissue culture? Because we have discussed in its application of the plant tissue culture that we are going to conserve or preserve some sort of species which are extinct. So by preserving or conserving those species which are extinct and which are not living today, so we have to conserve their germplasm, their genetic material as well. So now we are going to consider why there is a need of germplasm conservation and how we are going to conserve the germplasm of a particular species. So we can say that, in other words, as the tagline indicates, that it is the conservation of a genetic resources. Those genetic resources may be in any form, in any form, but it should be viable, it should be living, so that whenever if we need it, we can grow it and use it for our experimental purposes. So first of all, we have to discuss the germplasm. So what is germplasm? It is the sum total of all the genes which are present in a crop and its related species constitute its germplasm. So there are various definitions of germplasm. There is no clear-cut one single definition. We can define it in various ways. So for example, what is germplasm? In the first definition we have discussed, we have defined it that it is the sum total of all the genes which is present in a crop. So as for example, if we are going to conserve or preserve crop, for example wheat crop, or Cicer erytinium, that is the gram plant, it is also a crop, or any other crop which we are going to preserve it. So in that crop, all the genes, all the genetic material which is present, which is functional, which is functioning as a genetic material, so it should be. So sum total of all those genes, either they are in thousands, either they are in hundreds, or they are in billions or millions, so whatever the number may be. So those genes constitute the germplasm, and in its remember, and its related species. So remember, for example, we have a species, and below the species there is a rank of ecotypes as well, and there is a subspecies as well. So these are the related plants, related individuals, which are different in some extent because of living in the different habitats or due to some other reason, but but mainly when we focus them, when we consider their genetic material, so they are related to one another, they are almost the same. So it is not only the genetic material of that particular crop, but we are also going to conserve the genetic material of its related species as well, because if, for example, there is a species which is now going to extinct and we have to conserve it, so we are not conserving or preserving the genetic material of that particular species only, because somehow, because of some reason or the other, there is a possibility that it may be lost or it may not be viable. So we are going to conserve the genetic material of its nearly related species, because if we lost that genetic material of that particular species, so we can use the genetic material of these related species to somehow constitute that particular germplasm genetic material, and somehow we are going to make it again. So that is the definition of the germplasm, or it is ordinarily represented by a collection of various strains and species. So remember, there is a species level, and below the species level there is a strain and variants and varieties and cultivars. So these are the terms which are used below the species level. For example, in wheat crop we have hundreds and thousands of varieties of wheat crop. That is only the Triticum aestivum. The species is one, Triticum aestivum, but we have changed certain traits of that Triticum aestivum. So those traits are mainly beneficial for the human beings. So by changing those traits, either through the breeding practices or through some other way, so it changes some sort of the genetic material. That genetic material is not natural. So because of that changes in the genetic material, it will make a cultivar or variety, but the species will be remain the same. So here same is the case here with the strains as well. So these are the variants, we can say that variants of the species which are present in according to the which are modified according to the need or according to the situation. A germplasm is a collection of genetic resources for an organism. So in a very simple term we can say that it is the collection of, it is a sum total of all the genes which is present in the organism, and when we are going to conserve it through some one way or the other, we are saying that we are conserving the germplasm. As you have studied in the previous classes, that there are some biodiversity hotspots. There are many species which are extinct, most of the species which are endangered and nearly extinct. So there is a need to conserve their germplasm, because in the future there may be some process by which we can regenerate organisms like those, so that as these species have a very beneficial function for the ecosystem, so their conservation of the germplasm is very important. So plant germplasm is the genetic source material used by the plant breeders to develop new cultivars. So as far as the plant germplasm is concerned, when we have a germplasm at our disposal, we can use it in various ways, in multiple ways. For example, if we talk about for the in the context of plant breeding, so those plant breeders which are concerned with the making of the new varieties, making of the new cultivars, they use the germplasm for their own experimental purposes, and by experimenting and by carrying out experiments and on those germplasm, they can make a new cultivar. For example, there is a wheat variety or a wheat species which has a very low yield. Now what we do, we conserve the germplasm of that particular wheat, and we give it to the and we available that germplasm to the plant breeders. Now plant breeder will thoroughly investigate the genetic material, genetic resources of the species, and he or she will try to make or try to indicate those genes which are responsible for the yield, and somehow or the other, by the breeding practices or some other genetic or biotechnological way, he or she can change or manipulate those genes, and the wheat variety, the new wheat cultivar, it may have a better yield. So as far as the germplasm is concerned, it is not only used for this purpose, there are many other ways. When we go to the different fields of genetics, for example the biotechnology, population genetics, we have we can say that a classical genetics, so evolutionary genetics. So these are the various fields, various branches of the genetics, and when we study these germplasm in different fields, so their perspective and their purpose is changed accordingly. So as far as the germplasm is concerned, so from where we are going to conserve it, what is the source of germplasm conservation in the plants? For example, when we talk about the plants, as our topic is in the plant tissue culture, so we are concerned mainly with the plants. So those germplasm may be the seed. So most probably the source for the germplasm conservation is the seed. For example, we are going to save, or we are going to protect, and we are going to store a seed, for example large amount of seed which is available, but there is not the cases for every species that and the seed will be available. So there are some species in which the seeds are not available. So other plant propagules which can be grown into a mature plant. For example, there are some species which are vegetatively which can be grown. For example, we can say that in the Poaceae family there are sugar cane, and in the Solanaceae family there is a potato which has an underground stem, and that underground stem, the rhizome, it can regenerate and regrow and form a new plant. So in those cases, those potatoes and these stems which are able and which has the capacity to make a new plant, those also may be conserved. So they are also called the propagules. There may be a leaf, there may be a stem, pollens, as well as these all contains cultured cells. These all contains the genetic resources, genetic material which is able to generate a new plant. So that's why germplasm conservation, in other words, we can say that it is the conservation of any plant material which is able to regenerate and regrow the whole plant. It has the capacity to make a complete new plant. For example, there is a leaf, and we have discussed in the plant tissue culture that we can make a complete new plant from the leaf. We are isolating the cells from the leaf. There will be those cells will be cultured in a nutrient medium, and the callus will be formed, and from those callus we can induce a somatic embryogenesis, and by the somatic embryogenesis there is the chances of the production of a complete new plant. So either those cells may be from the leaf, they may be from the root, as we have discussed in the Dacus carota in the carrot experiment, that the cells were taken from the tap root of the carrots and they were cultured, or there may be the stem, there may be pollen or cultured cells as well. So the in situ and ex situ preservation. So as far as the preservation methods are concerned, there are two major or main methods. There may be those may be in situ or ex situ. What is in situ and ex situ conservation? We are going to discuss it further as well. This material is known as germplasm conservation. So either we are preserving those species. For example, or we can say that in situ conservation is the preservation of a genetic resources or the plants at their habitat. So when those plants are growing in their habitat, so they are conserved, they are preserved, they are protected from the damages. Ex situ preservation is the preservation outside their habitat, natural habitat. For example, if we are going to preserve them in the laboratory or in the pots in the greenhouse, so these are ex situ preservations. So in situ preservation and ex situ preservation, when these are either of these is used to preserve the material, that is known as germplasm conservation. And what germplasm do? Germplasm provide the new material which the breeder uses to develop commercial crop varieties, and we have discussed it previously. Preservation and conservation of plant diversity is an important issue. So what is the need of preservation? As we have discussed in the previous slides, and I have given the detailed account of the preservation method and why we are going to preserve it, but here we have discussed it separately as well. So preservation of the plant biodiversity. So as far as the plants are concerned, there are many species which are under the threat. So they are either extinct, they are either endangered, or they are near the extinction. So those all the species, they need to be preserved or conserved as far as their genetic resources are concerned. Storage of economically important, endangered, rare species and make them available when needed. So what is the preservation purpose? So when we store, for example, there is a species which is economically important but it is under the threat, it is endangered because of a very more or expensive use. So those economically important species may be stored. The endangered species which are under the threat because of the habitat loss or due to some other way. Rare species which are very low in number naturally, so those also should be conserved, and whenever we need it will make them available. So the conventional method of the storage, for example, there are methods which are used to store the seeds. For example, we can say that seeds of the crops and other species, but those conventional methods have various drawbacks and fail to prevent the losses and due to various reasons. So those storage processes which we have or which are followed conventionally or at the domestic level, they have a various drawbacks, and those drawbacks are now overcome by the using the in situ and ex situ conservation, by using the plant tissue culture or some other way. So as far as the methods of germplasm conservation is concerned, we have discussed it previously, that either that is in situ preservation, that is the preservation of the plants in their natural habitat. So how they are preserved? For example, we have various national parks at the Pakistan at the country level. For example, near to Dia Khan there is a Sheikh Budin National Park. Here we have various plant species which are preserved, which are conserved here. And Lal Suhara National Park is in Bahawalpur, and you can make a list of those national parks which are present in the Pakistan. So these national parks, and there are also other some categories which are related to the preservation of the species which are present in that jurisdiction. So in the national park, the species which are present there, they are preserved, they are protected, and they try to protect them from the harmful effects, from the damages, either they are catastrophic or they are anthropogenic. So they are protected from all the damaging effects. So ex situ preservation, that is, you can see that they are preserved in the greenhouse, they are present in the pots. So as they are present in the pot, they are growing in the soil, but they are not in their natural condition. So they are brought in the laboratory, they are brought from their natural habitat to a greenhouse, and now they are preserved here. So that this is the in situ and ex situ preservation. So we are going to discuss it further again. So in situ preservation is the preservation of the germplasm in their natural habitat where they grow naturally, and the conservation of domesticated and cultivated species in the farm or in the surroundings. For example, either we have made a farm where there are various species which are growing there, or there is a national park or some other way which is protected somehow. So protection is necessary. What does it mean? That those species should not be lost, or they should not be damaged by any other way. However, there is a heavy loss or decline of species, population and ecosystem composition which can lead to a loss of biodiversity. But due to rapid urbanization, rapid increase in the population of the world, there is a rapid urbanization. So there is a continuous habitat loss. Forests are now cut down, and there is a housing societies which are now present in those forests, and because of that habitat loss, the ecosystems are lost, and those plants which are present in that particular area, now they are not available, because various species, some species, they are very restricted to their habitat, what we call the micro habitat, and you will not find that species in a, for example, 1 to 2 kilometer area. You will find the particular species in a 1 to 2 kilometer area, and beyond that area you will not find that particular species. So as far as some plants or some species are concerned, they are very restricted to their habitat, so that there is a need to preserve their habitat. So due to habitat destruction and the transformation of these natural environments, in situ methods alone are insufficient for saving endangered species. So as we have discussed, sir, there is a rapid urbanization, there is a loss of the habitat destruction, there is a biodiversity loss. So because of these all the issues, so in situ preservation, in situ conservation, conserving or preserving species in their natural habitat, it is not a way that it is 100% efficient. So it is insufficient, and because of that we have made another step, and we have made ex situ preservation, and now we are preserving those species in a laboratory or in a synthetic habitat to maintain the biological material outside their natural habitat. So they are outside their natural habitat means the habitat may be in the greenhouse, in the glass house, or maybe in the laboratory, in the culture medium. So outside the habitat, the natural habitat means everything where it is growing. Storage in seed banks, field gene collections, in vitro collection, and botanical gardens. So these all are the methods by which ex situ preservation is carried out. Ex situ conservation is a viable way for saving plants for extinction, and in some cases it is the only possible strategy to conserve the certain species, because we are not able to conserve some species in situ by practicing the in situ preservation. Why? Because those species are very restricted to an area, and because of the urbanization, because of a certain project, there is a maybe a loss of the habitat, and now we are not able to grow that species in any other area in a natural habitat. So what we do, we brought that species in a lab condition, laboratory condition, ex situ condition, and we preserve those that species. So in vitro preservation or conservation is especially important for vegetatively propagated and for non-orthodox seed plant species. So there are another way that in vitro or laboratory preservation methods are very important, are very necessary for those species which are vegetatively growing. So those species which have a seeds, so those seeds can be preserved, but the species which have a vegetative propagation, so they don't have a species. So conservation methods, especially those, it is important for the species which have a non-orthodox seed plant species.